Okay, welcome everyone to Bioinformatics for Infectious Diseases. In today's session, we'll speak about the emergence and the development of bioinformatics and the application of bioinformatics in infectious disease research and applications. We'll discuss what infectious disease are, how they can be categorized from a data perspective, how could they be studied, and specifically we'll focus on next generation sequencing data. So what methodologies are there that we can apply to the study of infectious diseases? We'll also talk a little bit about some of the tools and resources associated with the program that you can leverage in between sessions and tackle some of the logistical questions that you might have. Get to know each other. So hopefully all of you here are ready to study bioinformatics and learn about infectious diseases. And so to kind of move on into the topics of today's session, we will go over some of the examples of bioinformatics analysis methods that are used in pathogen diagnostics, viral evolutionary studies, antiviral drug discovery, vaccine design, and the characterization for emerging drug resistance. And so we'll talk a little bit about different tools, different data types, and different methods, and that will get us started for today's program. But before we go on, let me tell you a little bit about us. So Pine Biotech is a technology transfer company that was originally developed at the University of Haifa. So they developed some techniques for methods, easy to use methods of bioinformatics analysis. And this was started by Dr. Alfred Tauber, who comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. And also Dr. Leonid Brodsky, who is the director of the Tauber Bioinformatics Research Center at University of Haifa, and is an expert in bioinformatics and biostatistics. Together with our team, what we do is really translate the tools and the methods that have been developed at the center and at large for the application of different biomedical and other types of studies to make them more user-friendly and to train a community of users how to use them effectively. So what are we going to study in this program? Bioinformatics for infectious diseases is going to be focused on the different types of data, resources, and tools that you can use to apply bioinformatics to publicly available data sets on infectious diseases. And so we'll talk and understand viral diversity and its role in epidemic infectious diseases that keep reemerging, including zoonotic transmission, transmission of disease between humans, and the process of viral and bacterial disease development. We will also learn about bioinformatics analysis approaches to study viral genomes, as well as bacterial and other types of pathogens. And we will talk about different strains, haplotypes, how to find differences in different sequences, and how to associate them with phenotypes. And as a result of completing this program, you will gain some hands-on experience, hopefully by analyzing curated data sets that we've sourced from public domain collections that were developed into training tutorials guided by experts with bioinformatics experience and knowledge about different aspects of infectious disease. So let's talk a little bit about the difference between infectious and non-infectious diseases. So an infection is the invasion of an organism's body tissues by disease-causing agents, their multiplication, and the reaction of host tissues to the infectious agents and the toxins they produce. An infectious disease, also known as a transmissible disease or communicable disease, is an illness resulting from that infection. Infections are caused by infectious agents that we call pathogens, and these can include viruses and related agents such as viroids, bacteria, fungi, parasites. And those parasites could be unicellular organisms like malaria, toxoplasma, babesia, or macroparasites like worms or helminths. So non-infectious diseases are also recognized as chronic diseases and can be caused by such factors as the environment, nutrition, and lifestyle, as well as genetic inheritance. The World Health Organization recognizes only 4 main types of chronic diseases and non-infectious diseases: cancer, cardiovascular, chronic respiratory disease like asthma, or chronic obstructive pulmonary disease, COPD, and diabetes. So the replication of pathogens in the host leads to major interference that starts from membrane disruption of an individual cell, as in the case of viruses, and the release of toxins, as in the case of many bacterial pathogens. Many of these interferences induce molecular biological pathways designed to fend off intruders and signal to neighboring cells of danger. The pathogens can also modulate these pathways and cause long-term organ damage, such as lung tissue scarring due to the immune hyperactivity. Hosts can fight infections using their immune system. Mammalian hosts react to infections with an innate response, often involving inflammation, followed by an adaptive response. Vaccines are used to train the immune system to identify pathogens and store the necessary epitopes for antibody production in the long-term immune memory. But due to a variety of factors, such as immune response, can be insufficient on their own. 2 main approaches that have significantly reduced death from infectious diseases in the past century are the use of antibiotics, antiviral drugs, and vaccines. So vaccines have been discovered first as far back as early 1800s and becoming major instruments to train the human immune system in response to various pathogens, reducing worldwide deaths significantly in the 20th century. As a result of water treatment, so some other approaches to kind of mitigate disease, infectious disease, the use of vaccines and antibiotics, the death rate from infectious disease was reduced tenfold in the 20th century. But the 21st century, as we know, came around with absolutely new challenges, including antibiotic-resistant bacteria, as well as novel pandemic viruses that we were not prepared to address, because previously most of the viral diseases that vaccines were designed for were chronic diseases. So they were infectious diseases, but they were around for a long time. So how is bioinformatics used to study infectious diseases? Bioinformatics can be used in many different ways to understand these pathogens and how they evolve. So one of the features that is unique about the genome is that typically the shorter the genome, the faster it changes. And so the accumulation of mutations leads to whole groups of novel pathogens, and those could be studied using next-generation sequencing. What you see here are ways to study evolutionary relationships to understand the emergence and trace the origins of diseases. We can also look at virulence, or what causes a change in phenotype, how one virus, for example, can be more virulent, more infectious. We can look at protein expression, and using those proteins design different types of molecules that can interfere with the function of those proteins. We can use diagnostic tools to monitor these kinds of viruses and pathogens, and really understand very specifically what type of pathogens they are. And we can also look at the population structure and evolutionary history by understanding the diversity of these rapid changes as they represent the responses to different environmental challenges, like the host immune response and treatments with drugs and vaccines. Genomics and bioinformatics have contributed immensely to our understanding of infectious diseases, from disease pathogenesis, mechanisms of interaction, the spread of antimicrobial resistance, to host immune responses. And as a result, the emergence of genomic medicine has substantially advanced our ability to detect, study, and treat infectious diseases. These achievements typically come as a result of a close collaboration between people that understand how to apply computer programming and databases, or computer scientists, and biologists, so those people that understand the biology of the infection. And this places bioinformatics at the center of many current advances in this domain. So for example, what you can see here is how we can apply different computational tools to extract signatures associated with the response to viral infection, how we can trace the lineages and study relationships between different types of pathogens, how we can then also track the specific mechanisms of action that are used by viruses to, for example, infect the cell and prevent the entry into the host, or how we can track the responses of the immune system so that we can understand better the different types of mechanisms and the modulation of these mechanisms by specific pathogens. Ultimately, we live today in the era of multi-omics integration. And so what we will try to do in this program is talk about different types of omics data, and those types of omics data could include genomic data, or the study of DNA and RNA variation, sequence variation. We will talk about transcriptomic data, which is the study of genes and their expression, the levels and the changes in their expression. We will talk about different types of protein structures, how we can study the protein, folded proteins, and look at their crystallized structures and identify different types of active pockets and different unique features of those proteins. And we will talk about the complexity of biological data that is represented by a growing interest in a more detailed view from the full host to tissue specific to cell specific, so single cell data. Now, omics data can be used to study viral genomics and evolution. So how do they dynamically change over time? We'll take a look at some examples in previous pandemics and understand relationship between the spread of a disease and a particular sequence change in the viral community. We'll talk about the microbiome and infection. So how does the community of microorganisms in our body can affect the infection and the spread of that disease? We'll talk also about immune response and biomarkers. So how can we detect specific changes in the host transcriptome to understand what are the specific pathways that are activated and what are some signature genes that we can trace to understand the difference between an acute response and a mild response to infection. One major way that bioinformatics is changing our understanding of the microbial world is our ability to ask basic questions based on data. And so because we have so much data available in the public domain, we are going to be looking at different types of repositories of different data sets that are available on repositories like NCBI, the National Center for Biotechnology Information. And we'll look at the different ways that people use sequencing to study different types of infections. Okay, sorry about that. Okay, does anyone have any questions? Okay, does anyone have any questions from what we discussed? So if you have any question, please write it in the chat, and we'll try to make it as interactive as possible so that we understand that the things that we're discussing are clear and there are no questions as we're moving along. So again, I'll just pause here for just a minute, make sure that there are no questions. So if you do have a question, just type it in to the chat box. Okay, let me ask you, how many of you, okay, William is asking, will be those tools based on free website tools? We will go over some of the tools just now. We're going to be transitioning to speak about next generation sequencing. So I'll introduce you to some of the tools and you can try them for free, of course. Yes. Okay, Chika is asking, what are databases or the differences between databases and repositories? Excellent question. So I think maybe a general distinction could be made between very different, so databases that are very specific. For example, there's a database called NCBI virus, or there's a database called Malaria Gen. So these could be general about viruses, or they could be even very specific to a particular pathogen like malaria. And there are very generic databases that are non-specific. And those databases include the repository called NCBI or ERA, right? So these are repositories that contain many different types of data sets that are non-specific. So I would say that those are some differences between publicly available data sets. Now, there are also databases that are private or controlled with controlled access. So GIS Aid, for example, is a repository where registration is required and approval of your account is required before you can actually access the data. So those are some of the databases that we'll be going through, and I'll just list them here in the chat for you as well. Okay, is it possible to analyze the viral transcriptome rather than or in addition to the host with this platform? Well, so as we speak about next generation sequencing and how next generation sequencing data looks like, you will see that the analysis itself follows a certain logic. And that's why we call these programs OmicsLogic, is once you understand the logic behind the analysis, you should be able to take the tools that you understand how they work and apply them in a meaningful way, depending on what kind of data you have. So remember that transcriptomics, or the transcription of particular genes, means that the sequencing was done in a specific way, and you have to map it on a particular reference genome with a certain level of quality control that you could apply to that process. So as we learn about these tools, you'll see that they are fairly universal. You can use them in many different ways. And once you learn how to use them, then I think it's up to you, or up to the data that you have, as to whether you can apply it to a particular type of a project or a data set. Okay, excellent question. So alternative splicing, can it be used in the analysis of transcriptome of a virus? So I think, again, one of the challenges here, as you think about viruses and how they replicate very differently. So for example, in the human, in the host genome, there is the specific gene sequence with exons and introns, and there are specific regulatory assisting enzymes that are present there to facilitate the splicing, right? And so alternative splicing has been known to differentiate between different cancer types, for example, or has been shown to be different at different stages of development. And so those kinds of alternative splicing events are well studied in hosts, in model organisms, in humans, and animals like mice, rats, etc. In viruses, I think that less of the mechanisms that are typically used for alternative splicing of human genes are going to be expected from viral genes. The virus especially typically leverages the host machinery to produce its proteins. And so I think it has much less control over how that alternative splicing is being done. And so that's why I think we expect a lot more diversity. Not only is that diversity is going to be between virus of the same type, but different strains, but also in different cells, in cells that are specialized to do different things in different tissues and different organs. And so because of that complexity and diversity, I don't think that as much has been studied and known about alternative splicing in viral genes, but I think that's something that you can definitely try and replicate, especially if you're using things like cell lines, but I don't think that as much has been studied. Does it mean that one has to use the host genome with respect to the virus? No, I think if you want to study the viral genes, you definitely have to use the viral genome. Okay, when we say data set, do we mean only the information obtained from the genome of an organism, or does it include its phenotype? And I think that's an excellent question, Chica. So many of the databases provide us with access to different types of data. So sometimes if the data comes from patients, the metadata or the phenotype about the data could be protected, and we might not have a lot of access to that. Sometimes we do have access to both the genomic data and the phenotype. So we'll take a look at some of the examples and we'll see. I think it differs from project to project. Excellent question. So thank you again for the questions. I think this is really the intent of this session today, and during the sessions as we go through, hopefully we'll have a lot more discussion. So knowing what is interesting to you and what are the questions that pop up as you go through the session is very informative. Okay, so next generation sequencing. Why do we need processing of next generation sequencing data? So the processing of next generation sequencing data is the process of extracting material, biological material, in a specific format using different types of library preparation techniques and sequencing them to produce some sort of information. And that is why we're going to talk a little bit about what are the logical steps to take this raw sequencing information, eliminate the noise that comes with the technical steps of data preparation, and then what type of information we're looking for as we quantify and analyze the data that we have. First of all, I have to mention that there's a huge number of data sets that are continuing to develop and to be available on different types of repositories. So it seems like the amount of data has been growing exponentially over the past several decades. And I think that especially in the recent times, we have seen an amazing number of genomes that are available across a variety of different domains. But I think this is especially true in the domain of virology, so specifically for viruses. And to understand that, we need to understand how are these viruses actually sequenced. So there are many different types of data preparation steps that could be used to be able to ask specific questions of the data. So data is not just generated by a fully automated process. That process has a design in mind. So let's talk about 3 different types of sequencing, and then we'll take a look at some of the use cases with those types of sequencing. One example is targeted sequencing. So a good example of targeted sequencing is if we are looking for specific genes that we know are associated with drug resistance, or specific genes that encode for proteins that are especially critical for viral entry, for example. Can someone think of some examples of those kinds of genes that could be sequenced in a targeted way and why would we do that? So why would we sequence them in a targeted way? Okay, any examples that you can think of? What do you think might be the reason, or what an example that you might think of to targeted sequencing of a particular gene? Okay, great. So we have an answer from Chica: genes coding for viral spikes or nucleocapsid. Right. So if we know that there is a particular protein encoded in the gene, and we want to understand the variation of that gene, we can focus the sequencing on just that one gene. The benefit of that could be that we get higher coverage, and so we can sample in a deeper way variation of that particular target gene. Another example could be with targeted sequencing of malaria drug resistance genes, right? So in malaria, there are thousands and thousands of genes, and we are expecting to find some single point mutations that are responsible for drug resistance. If we are sequencing a collection of pathogens, of parasites like malaria, then we're expecting to sometimes find those mutations and sometimes not find those mutations. And so that is where we will benefit from a higher depth or a greater depth of that sequencing. Ifioma is saying 16S for bacterial isolates. Excellent example. So when we take an environmental sample, instead of looking at just one organism, we're looking at many different organisms, and to get some meaningful information like their taxonomic assignment, which taxonomy do they belong to, it is useful to focus on the 16S ribosomal RNA gene to understand how this conserved gene can help us group the different types of organisms that we have in the sample into defined groups. And William is saying, target B protein from bacteria or epitope prediction. So if we have some specific regions of a protein so that we can find in the gene, we can also look for specific variations there. I think that is also one of the use cases. So that is a targeted sequencing. Now, targeted sequencing is typically for a specific sequence that we're looking for. Sometimes we're going to be calling this amplicon sequencing. So amplicon sequencing means that the library is designed to amplify using PCR primers. We're amplifying a particular type of a sequence, like the 16S ribosomal RNA, and then we're able to study only those types of genes. Now, this is different from the whole DNA and RNA sequencing. And one of the reasons is because whole RNA and DNA sequences are not going to have a uniform sequence. So the longer the sequence that we have and the more variation in the sequence that we have, we will have a variable coverage for that genome. And so those are some different types of sequences. So always remember that the type of information that we are getting is directly related to the type of data that we have. And so when we look for data, when we are trying to find a data set to work on, it's important to understand what are the specific techniques and methods that were used to present this data to us. And that will guide us in what steps we need to make to analyze this data. So here we have two examples, viral outbreak methods, and here we have what's called viral metagenomics or clinical metagenomics. So this is an example where from the same biological sample, you can actually prepare a full sequence, a consensus sequence of what we expect to find. And you can also find the specific viral genomes that you might expect, or haplotypes that you might expect to find, and that is called metagenomics NGS, or mNGS. Now we'll talk about these in greater detail, but let's also take a look at this side over here. Here we have an example of microorganism identification. For example, if I have a tissue or a sample that is infected with a virus, how do I understand what virus is it infected with? Maybe I need to map it on a whole database of potential organisms to identify the specific type that matches what I have in my sample. A lot of times microbiome, right? So as we spoke about the different types of environmental samples, when we are looking at a collection of microorganisms, we're expecting a lot of diversity, and maybe we know that specific types like actinobacteria or proteobacteria, right, are associated with specific phenotypes. So we can also try to analyze what types of bacteria do we have. And finally, you can see right here, transcriptomic analysis. So could we find some differentially expressed genes that are specific to a stage of infection or represent the difference between infected and non-infected? So those are some of the use cases, some of the applications of this type of sequencing. But also we have to remember that there are many different types of sequencers today. So it used to be that we had just one type of sequencing, but today we have at least 3 different platforms or 4 different platforms potentially that you can come across. And so when you're looking at data sets, that's another consideration. You need to remember that they could be produced in different formats, and that is coming from different types of sequencers or types of mechanisms. So here we have the PacBio sequencer. You can see it's a huge machine. What's unique about PacBio is that they provide a fairly accurate long sequence reads. We have the Illumina, different types of Illumina sequencers. They provide typically very high accuracy, but short sequences for the reads. And here we have Ion Torrent, and we also have Oxford Nanopore. So these are Oxford Nanopore, typically lower quality, but longer reads. And then Ion Torrent is kind of like a mix between the two. The technology doesn't yield as much data as the Illumina sequencers, but other than that, it is comparable. So how do we analyze such data? The raw data that is coming out of these machines is stored in different file formats. So these include FASTQ files, Fasta files, Gtf files, BAM files, SAM files, etc. So to understand how we're going to go from this raw data that was generated using a sequencing machine, we need to understand what is inside these files and what are they used for. So FASTQ files essentially contain the raw sequence reads. And this is important because the raw sequence reads were generated typically on a random, right? They are randomly generated. What that means is that all of biological material that we have has been first, we separated the raw genetic material, DNA or RNA, and then that material was shattered. And so we end up with these short sequences. And the short sequences are those reads that are going to be stored in the sequencer. The second format is Fasta. The Fasta format is typically used to store individual reads, but more often it is used to store full genomes or whole sequences. So for example, your reference genome could be in a Fasta format, or it could be in what's called a GenBank format. So the Fasta format just contains the raw sequence and major sections of those sequences. For example, if you have chromosomes, if you have multiple types of sequences inside the file, it could be separated by a line with this sign in the beginning, as you can see right here. And finally, we'll use the Gtf file. The Gtf file or the GFF file is genome transfer format. It is a format where we store the beginning and the end of annotation. So for example, if we have a gene, we know that this gene is located on chromosome 11 at position 100,523 up until position 121,234. So that information, including sometimes the name of the gene and the different transcripts of that gene, are going to be contained in a GTF format. Now, the Fasta file, as I mentioned, could be a way to store complete sequences. The complete sequences, for example, a full genome sequence. So here you have a Fasta file of a full organism, Spodoptera frugiperda, isolate SF9, scaffold 12. So a scaffold is going to be a portion of that genome, whole genome shotgun sequence. If you have a scaffold 12, that means you have scaffold 1, 2, 3, etc. So you have other scaffolds. Here you have a complete genome. So again, you have the ID right here in the beginning, the rabies virus, and this is a complete genome. Typically, for shorter genomes, they're going to be a full sequence in a Fasta file. For longer genomes, they could be broken up into parts, scaffolds or chromosomes. FASTQ file, here you have the actual sequence of the read, plus what's called the pH-read score. So this is a score coming from the sequencer of how clear the signal is when the sequencer is reading the actual chemical reaction that produces this image that it is capturing and translating into letters. So this is a process that typically is going to go downhill from multiple uses. And so every so many runs, the machine is cleaned to improve the quality. But sometimes we want to take a look at the quality, make sure that we don't keep the low quality portions of our data so that in our analysis, we're not analyzing noise, right? Because garbage in, garbage out. That is the destiny of bioinformatics. So how does this process work? We take the genomic information, the DNA or the RNA, that information is shared or broken up into fragments, and fragments could be, the length of those fragments is important. We'll talk about the length of reads and how it applies. Then those are connected with adapters to prepare the sequencing library. And so those adapters, here you can see pair end reads and adapters attached at the end of each read sequence. Then these are applied to flow cell where we have Pcr Bridge Amplification. So Pcr Bridge Amplification produces enough material for the sequencer to read what's in the sequence. And the reading is done by synthesis with reversible terminators. So this is an example of Illumina sequencers. And here you can see what those letters look like. So depending on the color, this is the result that you get, and the result comes in a digital format. So here you have an example of what that sequence comes out to be, and this is an example of a single read. What can you do with this information? So for FASTQ files, we can take the FASTQ files, but because reads are randomly generated from random pieces that were generated from the whole genome, the first step that we have to do is we have to map those reads to the reference genome. Mapping them to the reference genome allows us to look at abundance of reads in a particular place, like here, and that is called coverage. Coverage can also be represented by a measurement of depth, and the depth is essentially how many reads for a given nucleotide across the whole genome. And with Fasta files, we can also analyze those Fasta files. For example, we can perform multiple alignment, sequence alignment, to identify relationships between sequences and measure that in some kind of way. So we can measure it in terms of distance, we can measure it in terms of evolutionary distance or molecular clock, and assign a time to the distance between them. So we'll talk about multiple sequence alignment as well. Importantly, these sequences are not just complete sequences that all have very stochastic variation. They also have specific types of sequences that we're going to be interested in. One of those basic types of sequences are called codons. So as you remember, you have individual nucleotides in your sequence, and that nucleotide, those nucleotides represent amino acids. And so we can take trinucleotide sequences and divide up the whole sequence that we have into amino acid sequences. Why do we do that? Because from the genome to the transcriptome to the actual proteins, the sequence changes from nucleotides to amino acids, which then fold and form functional proteins. And so using codon mapping, we can now analyze not only single nucleotide variations, but also mutations that are responsible for some kind of functional change at the protein level. So we can perform nucleotide sequence, multiple sequence alignment, and amino acid sequence, multiple sequence alignment. And those will give us different types of information. So that will give us some sense of differences between multiple types of genomes at the basic nucleotide level or at the functional level with amino acid mapping. How do we measure relationships between sequences? One way to measure a relationship is to look at individual sequence changes, so positions where we observe a mutation or a variant. Another type is if we want to globally look at similarity between sequences. So if I have one sequence that has, let's say, 10 mutations from the reference genome, and another sequence that has 20 mutations from the reference genome, I can say, well, these 10 and these 20 represent some kind of a distance from the reference. And now if I organize all of my sequences pairwise, I can start thinking about how do they form a sort of a relationship that I can analyze. And that could be analyzed with the view of evolution, because we know that the source of these mutations comes from adaptation and time. That's related to time. And so we can assign some kind of an evolutionary time to measure the change in sequences from an ancestor that, for example, we know or we might not know, and to attach it to the present day species or the data that we actually have. So let's try together to perform such analysis today and see what comes out. So to do this, what we're going to do is we're going to go onto a server that we're going to be using throughout this program. And the server, the account for the server comes with registration. So that's why I asked you all to register today. So if you could please go with me, for those of you that have registered, please go with me to server.tbio.info. Okay, I'm going to paste the link here. Once you go in, you should be able to log in using the same credentials that you used when you signed up. So when you signed up, it's important that you use your email and you created a password. So if you have an email and a password, you should be able to sign in. If you don't have an email and a password, let me know and I'll be able to help you. And once you do sign in, please put that you've signed in the chat box. Okay, do let me know in the chat once you're signed in, and we will go through this exercise together. Okay, so Harris, how did you, yes, if you signed in, So for example, here, this would be my email address. Okay. If it is incorrect, then I'll be able to send you the details. So at any point in time, okay, so those of you that have created an account today, yes, it should be correct. Harris, I don't know why yours wouldn't be working. Maybe if you have some additional, so I see Tori, Farrow, and Harris are having trouble signing in. I will email you your details after the session, so sorry about that. I will try to do it as soon as we're done. Okay, so if you are signed in, you will see here different areas of analysis. And so today we're going to do one of these examples just so that everybody can kind of navigate your way around this interface and learn about how this works. And if you have any questions as we're going through, please hold on to them. After this, I'll pause and we'll do a little Q&A session. Okay, so what we'll do today first is we will go right here under phylogenetic tree, and don't click on the title itself, click right here on this drop down button. And inside this drop down button, you should see a few demo examples. So you can take any one of them. I'm going to take this demo, Ebola phylogenetic analysis. So this is going to be an analysis to help us understand relationships between complete genomes of a virus. All right, great. So for those of you just signing in, again, where did we go? We went under here, data integration and modeling, and to this first demo, Ebola phylogenetic analysis. Once you get into this section, you should see that the start button is darker and the rest are kind of lighter. So what you can do is you can click on the start button. And notice that in each pop-up, you typically will find some text that explains this step. And then once you've selected that step, your next step is again going to be darker. So it's going to be highlighted what to do next. So the first thing that we want to do is we want to perform multiple sequence alignment. So multiple sequence alignment is based on this idea that we have sequences of whole genes, or a whole genome, sorry. So we're looking for global alignment between pairwise sequences and iteratively go through all of the possible pairs to make sure that we can either identify a change, so a substitution, or an insertion, or a deletion, and come out with a fully aligned Fasta file from the Fasta files that we uploaded, where we have gaps represented by dashes, and all of our sequences are exactly the same size. So that is the first step, right? So multiple sequence alignment, and then right here on top, click on OK. Once you've done this, remember I told you about nucleotide sequences, and then also we have amino acid sequences. So we can do alignment in amino acid sequences, and amino acids have different physicochemical properties. So these properties are useful because we can now look for those changes to understand how they are represented in the potential protein that is produced from this genome. And then here we have select codon position MSA. So actually, when we analyze the typical mutations that occur in different positions in codons, we know that it is not the same rate of mutation for every codon position. And so sometimes this is referred to third codon position or 4D position, and you can see here that this is only taking that specific position in the code. Now we have here different types of phylogenetic analysis methods. These include the standard ones, including in a R package called APE, or there is a different package called BEAST, which is Bayesian analysis of sequence that has different processes that are used to model this phylogenetic or the evolutionary clock, essentially. It's a way to model the evolutionary clock. And to compare all of them, we can just click on all phylogeny methods. This will include all of these methods that are listed here. And once we've generated a phylogenetic tree with many of these different methods, we want to compare between them and look at the output as a tree space, essentially, that we can also analyze for consistency and select the best one that is most representative of what we think is the true biology behind what we have. Okay, did everyone create the same pipeline like me? Did anyone have trouble creating a pipeline like what you see on the screen? Okay, so Chica, if you are lost, let me know, where are you lost? Which stage are you at? Did you start? Did you have trouble finding this section in the main areas of analysis? Okay, at the start point. So Mohammed, if you don't have the password, it's the same password that you used for Learn. Okay, so the exact same password that you used for Learn. When you sign up for Learn, you get a demo account for this section as well. Okay, for Chica, just briefly, I will explain. You start, create a multiple sequence alignment of nucleotides. Those nucleotides are mapped using the GenBank file. They're mapped to their amino acid sequence. Then we analyze variation at different positions inside the codon and use that information to create a phylogenetic tree. Now we're using that multiple different types of models, and then we're producing a single output that is analyzed using a tree space to identify the most probable tree that is representative of what's happening in the data. And so to understand better the results, you can click right here on Download Pipeline Output Files. And here you will have a number of different results that you can analyze. So for example, you can look at all your trees. You can look at one of the trees based on one of these types of outputs. You can look at a table of all of your positions that compare the different positions at the nucleotide and amino acid level if you're interested in specific mutations that you're looking for. Or you can look also at a Fasta file of all of your aligned sequences. What type of information does this really give us? The type of information that we are seeing is a tree of relationships where the x-axis is the evolutionary time. So whatever we see earlier in time is going to be kind of closer to the left side on the x-axis. And we have a split that is represented by a potential common ancestor. And so therefore we can start grouping this phylogenetic tree and try to see association with the phenotype that we have in our input data. Okay, so we're going to be talking a lot about what kinds of projects and what kinds of data sets we can use to practice this. But here's, in this example, the phenotype that we have is location, for example, Gabon, Liberia, and Great Britain. And we have the date, 1996, 2014, and 2014. And so looking at this, we can see that the major difference between this group or this clade of the tree is that all of them are from an earlier time, including the reference genome and the early time sample that we provided as a baseline, and the later time. And this later time is also separated by location, but primarily it's separated by time. So as a result, what we can see is that this pipeline quickly allows us to see some major separation into groups. It allows us to understand how sequence variation is aligned with a molecular clock, and that molecular clock is correlated with the time that we have, but potentially we need the scale. And then it can help us start asking questions. And those questions could be much more specific about the specific nucleotide changes that are different between these two groups. Okay, if I go to my profile, I will see this is the program that we're in, Bioinformatics for Infectious Diseases. Okay, and here you can see some of the sessions that are planned. So we're going to have one of these synchronous, one of these in-person sessions a week. And in every one of them, we're going to be doing some kind of an overview of a topic like next generation sequencing today, file formats, right? Finding different patterns and things like that. And you will see a recommended course. So between now and our next session, the recommended course is genomics. If you just click on this right here, you will be taken to the course. And you will see that inside, there are some practical things that you can do. So sometimes you will see this tag, learn. This is theoretical material about DNA structure and variants. Then we will go into working with DNA sequences in R. How do you load sequences? How do you align them? And how do you display the alignment? Then we will go through an introduction to genomics, again, a theoretical course. You will find a course in R. You will find a course making a pipeline for a particular type of a data set, performing phylogenetic analysis, comparing sequences, and then also, so as a result, you will acquire skills about the logic behind the different data sets. You will get the idea of different types of pipelines that you can run for big data. So some of these data sets that we'll use are going to be gigabytes and gigabytes of data. And so whole pipelines are going to take maybe tens or hundreds of gigabytes. And so obviously it's impossible to do it on your laptop. So you will have our server to practice those. And then some of the smaller curated data sets, you will be able to work with in R right inside of our server. So let me give you an example. So here you can see one of these courses in R with an explanation of the specific script and with a practice. The practice starts with very simple assignments that you can do. So you can, for example, just click run, and these two sequences that are loaded here as an object are going to be aligned. And once you get the alignment, you'll also get a score. And so this kind of practice will help you understand how this type of analysis is done step by step, looking at both the global objective, like doing a pipeline that we did today, or performing the analysis in R and viewing the actual output so that you understand the technical part as well. Okay, so this is the course that is going to take you to our next set of topics, which are going to be related to the specific information in this alignment and how to build a phylogenetic tree that you can analyze and study. Okay, let me address some of your questions. So for those recommended courses, do you need to pay before access? If you have registered for the full program, so as you saw right here, once you're registered for the program, you will see what I see. So you will see your account and your progress, and you will get access to the Zoom links. And once the session is complete, you will also see the video recording right here and all of the associated courses that are down here. Now, if you've paid for the program, you don't have to pay for anything else. Some of you are LBRN. So LBRN has provided you with a full scholarship. You don't have to pay for anything. And you've gotten access to all of this for free after today's session. So those of you that were in today's session and gave me your account links, I will add those accounts to the program. If you have not registered for the full program, you can access some lessons in each one of the courses for free, but some of the assignments are going to be paid. So the basic subscription is only $10 a month. I think anyone can afford that. And so you can either go for that asynchronous access, or if you want to also participate in sessions like today, you have to purchase the full program license. Okay, for those of you that have a bad internet connection, so you will get a recording. The whole session was recorded. I will share the link, and the link is going to be right here instead of the Zoom link, you're going to see the recording here after the session is over. Okay, thank you, Tori. Again, for any one of you that joined maybe later in the session, especially for those of you that are from LBRN, and so LBRN is paying for your account, I need your URL IDs, just like Tori pasted right now, so that I can add you to the program access, and you will have access to all of the resources that are required.